Okay, welcome you guys. Welcome to the 12th Annual Minnesota Neuromodulation Symposium. I am Netoff. I am the conference organizer and also the director of the Center for Neuroengineering. I'm so excited to have you all here. It's going to be a fantastic conference. And if you have any problems, please come to me and let me know. We'll get it figured out. And I want to have the privilege of introducing Matt Johnson, who's our conference chair this year, who did an excellent job and just so thrilled about the whole program. So thank you, Matt, for everything you've done in organizing this.
Great. Yeah, thank you, Tay. So welcome, everyone. Happy May Day. We have a great program for you. This year, the theme is personalized neuromodulation. And in a sense, neuromodulation has always been personalized. It's kind of intrinsic to titrating neuromodulation therapy. But I think what you're going to see throughout today and tomorrow is the idea of bringing in anatomy, bringing in physiology, bringing in sort of patient preference into how we treat various conditions, brain conditions, peripheral nerve conditions, spinal cord conditions. So it's really exciting. It's a great lineup of speakers. Super excited to have you all here. Big thank you to Taylor Lopez-Angel, who's, I think, outside within the registration booth. She did an awesome job organizing behind the scenes. Big thank you to Taylor. Big thank you to Andrew Monk, who also helped out. Big thank you to the IEM staff for putting on this, and big thank you to the AV team. It's a real team effort to put this on. We'll also have a big thank you to all the sponsors. You'll see those, I guess, on the slides as you go through. Please take a moment to go out and visit the booths. Take some time to talk with them, see what kind of products they have, see how they might be able to collaborate with you in your own sort of research program. And also, please network. There's ample opportunities that we've provided through breaks to kind of meet outside. Try to meet someone new. Try to, there's a lot of people here that from around the country, and it's great opportunity to kind of meet somebody new and maybe spark that next collaboration. So a couple other logistic items. The restrooms are out the doors and to the right by the escalators. We'll have coffee and water kind of throughout the day, so can keep that going. And I think with that, I'll turn it over to a welcome address from Dr. Jerry Vitek. So Dr. Jerry Vitek is the McKnight Professor and Chair of the Department of Neurology at the University of Minnesota. Dr. Vitek is one of the nicest people that you'll ever meet. He's been a mentor to me. He's been a mentor to many people probably in the audience. He's the PI of an NIH-funded Udall Center, one of 3 in the country, and he is the new MnDRIVE Brain Conditions Chair. So thank you, Jerry, for giving us a welcome address.
Well, thanks, Matt. I always say Matt's one of the nicest people around, so that was nice of you to. I'm not sure everybody would agree with that, but that's okay. Okay, well, again, you've already got the welcome, but welcome to the 12th Annual Neuromodulation Symposium here at the University of Minnesota. This has been going on, as you see, for 12 years. It was started by Ben He, who's now in Pittsburgh, but the group here has done a great job in carrying it forward. So again, you've already seen the organizers and sponsors, but you should definitely get an opportunity to talk to the sponsors and talk to the organizers, talk to Tay, talk to Matt, talk a little bit more about the program. You had suggestions for years coming, but I think everybody would like to hear about that. I want to talk a little about the state of Minnesota and MnDRIVE. I don't think most of you know what that is. Well, what it is, it's an initiative between the University of Minnesota and the state of Minnesota, and it aligns the areas of research strengths at the university with the state's kind of key and emerging industries. And the whole goal here, it's been a really great union. This has been going on for about 14 years. We wrote that proposal in 2013 when President Kaler was here. And since that time, it's really been a great resource for those of us who have been interested in doing neuromodulation. What it stands for is Minnesota Discovery, Research, and Innovation Economy. And the mission for brain conditions, there's really five things here. There's basically brain conditions, there's global health, environment, there's clinical trials for cancer. The brain conditions was initially called neuromodulation. But because people felt, we don't think everybody in the country is going to understand neuromodulation, the people in rural Minnesota, for example, we changed it to brain conditions. Everybody's going to get that one. And the goal was to build our capacity at the University of Minnesota for world-class neuromodulation research. And I think it's been a really nice experiment in many ways, because it's been largely multidisciplinary across BME, neurosurgery, neurology, neuroscience, imaging, could go on and on. It's been a great multidisciplinary endeavor, and I think it's been successful. I'll show you why. The question was, why do we want to do this? Why did the state want to do this? One is because what we wrote in our first application when Tim Ebner and I put this in, Tim is a chair of neuroscience since retired last year, was that neuromodulation is one of the most exciting things that we've ever done. And if you've done cases like this, if you've ever been someone in the OR, when you watch them program a patient, the minute you turn on that stimulator in the operating room and that patient's tremor stops or they get more mobile, there's nothing like it. And we've done thousands of cases, but I have to tell you that each one is unique in itself, and you never get tired of doing it. So how do we do it? And I just mentioned it. The innovative treatments, brain imaging, neuromodulation, combining experts from across multiple disciplines together. And this is really kind of what helped us in a unique way to get the UL Center, because we have so many good people involved. What my mentor always told me, Mayo Long, before he passed away, is said that if you just surround yourself with good people, you're probably going to be successful. So what are the achievements that MnDRIVE has had? So if you're going to do this, align yourself with industry or with a company or with the state, you're going to have to show them benefit. And what we've been able to do in MnDRIVE, and I talk about the committee, steering committee, as well as what Dr. Ebner was able to organize when we first started, and what we're trying to do in terms of carrying it on is we've got about $237 million of additional funding that are related to MnDRIVE efforts. It's been a major part of us being able to get a UL Center, in my opinion, as well as multiple U grants and R awards. And over the years, it's provided care to well over 10,000 patients. In addition to that, it's really been a major help in recruiting faculty. So we've recruited 16 faculty in 10 years, actually 17. I can't talk about the last person because they don't want their name out there yet, but we have 17 now. It's also training the next generation of researchers. You can see here the number of fellowships that are offered through MnDRIVE, the number of trainees, the North Star STEM Alliance, Early Faculty Career Development Awards, and these are all young people that are working in the field of neuromodulation. So it's been a really, really kind of a big deal to be able to fund folks like this. We recently just funded another 4 fellows, another 6 graduate students, all for their research endeavors in neuromodulation. So you're going to talk about a lot of new things in the next couple days. New novel things, new ideas. But I'm going to take you back. I'm going to take you back to 500 BC when it first started. And I don't know if you know this, but the earliest use of electrical stimulation was back with the Egyptians when they used electric fish to treat pain. We're still trying to treat pain. We don't use fish, we use something else. And then Giovanni Aldini, who's a professor of experimental physics, he attempted to treat insanity with static electricity. And what's more interesting is I started delving into this, because it's kind of fun to get back into the history. He actually did some reanimation experiments. He used a volcanic pile, which is copper zinc plates separated by cardboard with some water between. And he would use stimulation on dead bodies, freshly dead bodies. And he could reanimate them. And you can see from the picture the impression that his colleagues had when he was animating these corpses. This led into things that J.L. Poole did in Columbia, and he attempted to put a probe in the caudate nucleus to treat depression and Parkinson's disease. And then in the 70s, there's also development of the technology. And you can see Hosobuchi, Richardson, and Keel, they're all doing things related to pain. So this is a long history, as I said, back to 500 BC. But what really changed things a great deal is when this man, and if you don't know who this is, you should know if you're doing neuromodulation. Alim Louis Benabid is a neurosurgeon. He was in Grenoble, France. He also had a PhD in physics. Those usually don't go together, but in this case, it does. And what he was doing, when we do lesion surgeries, which is how a lot of this started, you take a radiofrequency probe, you put it in the target, you get a lesion, you stimulate through it, you pass current. And if they have tremor or they're slow and their bradykinesia gets better or their tremor gets better, you say, okay, I've got a good spot, I can lesion here. But they would stimulate first to make sure that they had an effect. So you don't want a lesion, but you don't get an effect. And what he said is, if I can stimulate acutely and see that this is effective, why can't I just do this chronically? And so they collaborated with Medtronic, and they developed the first stimulation devices. And in 1987, he published VIM stimulation-free central tremor. And that had a big, big impact on the field, as you can imagine. And then he moved on to Parkinson's disease. And he did STN stimulation for Parkinson's disease. And everybody in the world started doing this, or trying to do it. It's not necessarily straightforward. But since that time, since this renaissance created by Dr. Benavid, whom I think should get a Nobel Prize, I think he thinks so, obviously, but I think he should too. He hasn't yet. I hope that he does. Going forward, what are our advancements in programming technology, our advancements overall? I'm just going to list a few and then just end up with a couple of quotes. So this is the old programmer. Anybody ever see this? Matt's got one, or Matt's seen this, right? We actually use it now in the Monkey Lab. But this is a dot matrix printer, if you can imagine. I remember carrying that bag around, or that box around, that suitcase they called it, going back and forth in the clinic. It was the first programmer we had. Now you got these things on your iPad. Now you can do remote programming on your phone. And we can go into these other advancements. Here's just a whole list of them, right? The physiology, understanding brain networks, that's all critical. We're getting much, much better at that. There are a lot of new technologies that we can use in doing that work. Surgical planning has changed. Our imaging, targeting, looking at parcellations of the targets we're going to implant in to know that we're getting into these motor areas or limbic areas or associative territories. What role does that play? If we get a lead that's too far outside the motor territory, can we still get benefit? Are we activating fibers? What are we really doing? The lead locations, hardware, new leads, directional leads have come out. There are different contact configurations. IPGs, pulse generators, now you can sense. Adaptive DBS has come out. There's imminent burst stimulation, different patterns, coordinated reset that can induce plasticity, the work that Peter Tass brought to the table. And then there's all kinds of alternating patterns people are exploring as we go forward. Programming software. What about remote programming? I just talked about that. You can sit in your office at home and program someone in their office wherever they're at. Now, there's some issues related to billing on this that people are trying to work through yet, but still, this has now been enabled. And I can tell you, when I was back in Emory in the 90s, late 90s, I remember talking to the CEO from Medtronic and said, you know, you guys do this for cardiac. Why can't we do this for DBS? He says, oh yeah, we could do that. So this just came out a couple years ago. So that's like 20 some years after I was told, oh yeah, no problem. Things don't move quickly in the field, sometimes. The other one is work with Boston Scientific. Now that was an Abbott system. Boston Scientific has something called Illumina, which is a very unique programming system. You move your current field where you want. You draw it out and say, I want to move it here. And they set all the stimulation parameters for you. And in talking, you're going to do some programming sessions, I think just after this, but talking to Leo Almeida, who's used this program, he said, there's no way that I'm going to figure out the constellation and combinations of stimulation parameters to get what they got. So there's all kinds of things that are going on. Now, algorithm-based targeting, pathway targeting is something else that's coming up. So this is, I want to move through this pretty quickly. But this is something that Cameron McIntyre gave me, well, maybe a couple years ago. And what he's trying to show here, if I can, I don't think I can advance it. Can you advance that video at all? Anyway, you're going to see pathways, and then that's going to get superimposed on an MRI scan, and then you can see a lead going into the target. This is what we're going to. We're going to have pathway activated. Not only are you going to have pathway activated programming, but you're going to have this patient-specific anatomy to work on for targeting. I'm going to leave you with 3 quotations. These are helpful for me. I'm not trying to give anybody advice, but I have to think about these things all the time when I go to meetings, and I've learned it over the years that, 1, pride is concerned only with who is right, but humility is concerned with what is right. The last thing is on wisdom, well, the second to last. And this is by Abhijit Naskar. He says, the highest wisdom is to know that you know nothing. And what I would say, my modification is the highest wisdom is to know what you don't know. In that case, I think I know a lot. The last thing I want to show you is this quote by the Dalai Lama. And for those of you who live to work rather than working to live, I think this is more important for you. And a lot of us could take advice, I think. So the Dalai Lama, when asked what surprised him most about humanity, he answered, man. He said, because he sacrifices his health in order to make money, then he sacrifices money to recuperate his health. And then he's so anxious about the future that he does not enjoy the present, the result being that he does not live in the present or the future. He lives as if he is never going to die, and then dies having never really lived. So I just saw this last night, which is why I wanted to put it in. I think a lot of us spend a lot of time working, lab, your research, but I think you need to find time for other things. Some things that actually make you more creative in the long run if you have a little time off. So take advice from the Dalai Lama. Thank you.
Great. Thank you, Dr. Vitek. So it's my privilege to introduce our keynote speaker for today, Dr. Kai Miller. Dr. Miller obtained his MD and PhD at the University of Washington, and like Dr. Vitek said about Professor Benabid, Dr. Miller has a PhD in physics, like Dr. Benabid, and an MD, and also another PhD in neurobiology and behavior. Dr. Miller is a pediatric and adult neurosurgeon at the Mayo Clinic. He specializes in treatments for brain tumors, epilepsy, and other brain conditions. He's been highly innovative in his work with dense array recordings to really understand electrophysiological dynamics within the brain. It's a real pleasure and honor to have you here, Dr. Miller, so we'll have you take it away.
So this is the first time somebody has invited me to be the keynote speaker of a conference, so I'm going to apologize ahead of time. So these are really smart individuals, but maybe not smart in this case because they invited me. So what I'm also going to do is try to be a little bit provocative and talk maybe about the arc of the history of the work that I've been able to be involved with. But I'm going to be omitting, I think, a lot of the really interesting work that we do at Mayo. And the reason why I want to tell you guys about that is because there's a lot of the people here from that team, and I won't be showing their work, but it's really interesting stuff. So you'll see it in later sessions. And I'm sorry, guys, you do wonderful work, and I hope that, that people get to see that throughout the time here. I guess the other thing is what Mayo is great at is clinical care. What we don't have is we don't have undergraduate, let's say undergraduate or graduate physics. We don't have the resources that, that I needed to get the skill set that I use day to day. And so the state of Minnesota does have these really nice programs to encourage collaboration between the two institutions. And Dr. Johnson and I were lucky enough to be supported on a grant for the last several years, collaborating together. And so that's really nice. And my hope over the arc of you know, the next 5 to 10 years that I have left in my life is to spend it working some with the University of Minnesota here. So while we're trying to get the AV set up, maybe I should just talk a little bit about the introduction that I'd like to have. So there's going to be a lot of words at the beginning of the talk anyway. But many of you see brain-computer interfaces in the news regularly. You see these fantastic stories where you have people, did we actually try to, well, I'll let you work on it.
It's not talking, so I think I know what's wrong.
Okay, so you see fantastical stories where people are generating synthetic speech from high-density arrays of electrodes placed in specialist areas in the brain, in early speech areas, in primary motor areas. But there's a few things that you don't know about this. So the largest, you know, and I hope to insult some of you, but only lightly to the point where we can have discussions. But the largest study that's been running as a clinical trial, and actually an FDA clinical trial for more than 20 years, uses these electrodes that are 1 to 200 electrodes, depending upon how many arrays they put in, and they get implanted in the brains of people who have disabilities. And I've been seeing these stories in the news, and I thought, well, you know, even though these things may not be scalable, and we'll talk about scalability a little bit later, these few individuals at least are getting great use of something that requires a whole team, but there's a lab set up in their house, and they're getting to do cool things with it. And then I found out that actually in the whole history of this program, no one's had open use of the device. So when they're not actively participating in experiments with these teams, they're not getting any therapy from these devices. And so you have to ask yourself, who is that benefiting? Well, if you hear the arguments of the patients involved, and I say patients only because they have conditions, they're not patients in these studies, they're volunteers for these studies because they're not getting a therapy. What they benefit from is the attention that they get. So they're not getting a therapy that they then use for engagement in the world around them in ways that they couldn't otherwise. So if if I had been, let's say I had had early ALS, I would be less concerned with participating in experiments and more concerned with being able to surf the internet in the privacy of my own home, especially when I was a younger guy. So the thing that I think we really need to be thinking about if we're going to create brain-computer interfacing is how do we create the kinds of devices that are going to help people? How are we going to create the kinds of devices that address a need and can be scalable, and which individuals are these actually going to benefit? So it looks like it may be registering there. Okay. So I see it down there. And to that end, what my talk is going to primarily be about is principles that we may need for brain-computer interfacing and some attempts that we've had for this. So I'll get started now. So the first thing, I guess, is I don't have financial relationships with ineligible companies. I think one of my sort of personal heroes and mentors, a guy named Kelly Foote, and some of his colleagues are here, said to me at one point that he'll take money from anyone that'll give it to him. I think I'll get there at some point, but I have to have enough different people giving us money so that basically you're equally conflicted amongst all of them. So the industry folks here, particularly from the fantastic neuromodulation companies that we have here in Minnesota, Mayo would be very happy to take your money, and so would I. And I'm not really going to be talking about off-label use of devices other than a little bit of work that other people have done. So when people say brain-computer interfaces, it can mean a lot of different things. For the purposes of this talk, what I mean is really something that does electrophysiological sensing. Okay, so there's and electrophysiological sensing from implanted devices. So I'm constraining what I'm talking about for brain-computer interfaces at the outset here, where we record an electrophysiological signal, voltage, and we have a variety of ways maybe of decoding this, and we can do things with that like control a device. So it could control a cursor on a screen, maybe a robotic arm, something like that. And I think what's equally important and infinitely more usable as we move forward are going to be the kind of devices that do brain rehabilitation. The total market for people who need to replace a full set of lost function by having an exotic implant put in their head is not that many people. There's not a market to sustain that. But there is a large market of people who have stroke and psychiatric disease and these kinds of things where we'll be recording and doing stimulation in tandem. So I've already given you a little bit of my soapbox while we're waiting to get set up. But now I'm going to get back on it. A lot of brain-computer interfacing and neuromodulation that you hear about, people are talking the most about, let's say, supremacy. So our particular milieu of research that we do gives us a 2% higher bit rate than other people had, and that's why we're in Nature and front page of the New York Times. Or it's like what you've seen before, only now we have an avatar where you can change their hair a little bit. And this is a slight exaggeration, but only a slight exaggeration for what goes out. Or we have the best scale of electrode or amplifier algorithm, and that's why we're really good. We're in Nature, we're on front page of the New York Times. But this really restricts progress, right? So if one study doesn't follow from another and there aren't shared understandings that emerge from the research that we do, we can't build future devices. If you want to start from scratch, somebody has a phone that's going off here. Hopefully I can get that to you. There you go. So we want to have studies that follow from one another. We want to have some principles we can use, okay? So I'm going to skip past this stuff. Sorry, Michael, one of my grad students put together some of these slides, but in the interest of time, I'm going to move past them a little bit quicker. Michael Jensen, he did a lot of the BCI work that we may talk about. He also put together these slides. But these are the kinds of things that you see, okay? And these are basically all Nature publications. Many of them resulted in, oh, we got a little bit of audio there, but resulted in really a lot of publicity. But what can we take away from this that's usable? And what I found is that maybe not a lot, right? So this is first to use an avatar, first to have a drone, or algorithms that are very exotic in the outcomes, and you have very high, let's say, very sophisticated demonstrations that you see, but the algorithms themselves are not transparent enough methods to reconstruct them. So I find things like this quite difficult to reconcile. The other thing is that most of the brain-computer interfaces that you see really involve this setup where you have a craniotomy that's done, and so craniotomy is an opening in the scalp, big opening in the bone, and you expose a large portion of the brain, and then you put these high-density arrays in them. Well, these arrays, the maximum signals people are getting out of them last for only a couple of years, okay? And you know, having a pedestal sticking out of your head in an exposed craniotomy, most of the talks that I go to, when you see neurosurgeons talking about this, they say this is relatively safe and it's easy and all that. I wouldn't do this to somebody, you know, and I think it's good to have a conversation, and if we have other neurosurgeons in the room who have or would, let's have a vibrant discussion afterwards. I wouldn't do it unless they're getting something out of it, okay? I wouldn't do this I don't do this with devices. If people have a tremor that's not that bad, I say, well, I wouldn't do this to my father. I'm not going to implant it in you. So why are we doing this when these people aren't getting open use? These participants that we have, I've already talked to this a little bit, talked with you about this a little bit while we were getting set up. But basically, these are not therapies for the volunteers involved. They just find people who participate who happen to have impairments. And the best justification that I've seen for that is that, well, if you destroy these areas of the brain where you're putting the arrays in, you know, if you put this in arm area, they're not using their arm anyway. Now, there may be a few exceptions to this, but it's not part of the largest studies. And the one thing I would say about Neuralink, we could sit and criticize a lot of Neuralink, but they're trying to have signals that last longer by putting in thinner filaments, and there also seems to be some home use because they're getting criticized for people using them for nothing more than video games. But if you think about it, at least people at home playing video games by themselves are getting something that they want out of this device. So I think that's actually a good thing about Neuralink, although I don't know how something that has received that much money, you could pay for the entire market of people who would potentially have a device like that with that money. So I don't know how they're going to recoup any of that, but that's a separate issue. And the question is do they know about the morbidity? Do they know how large the craniotomy is? I have patients who don't want to have their head shaved to have stereo EEG electrodes put in or DBS electrodes put in, so I don't shave their head. But you know, are people getting an idea for what they're undergoing, for the footprint that this is going to have on their head? You know, especially if they have lost some function, but they're relatively highly functional, which some people are. Be a lot of morbidity. That's big scars that you have. It's significant defects, and this is a set of experiments you're participating in for a couple of years. I'm going to skip past this because of time. I do think there needs to be a clear trail of discovery for scalable principles, meaning things that can be used for robust devices over long periods of time. And there needs to be less flash in how we do this and more basic statements of principles that should guide other people, where it won't be our Nature paper, it'll be their, you know, whatever, it'll be their Journal of Neurosurgery Impact Factor 3 paper, but one that actually guides patient care in the future. And we need to focus more on things that actually help patients. And one simple example for this is, you know, we actually know what a lot of patients need, and different patients will all need different things, but a lot of patients use assistive technologies and have been using them for decades. There's assistive technology for communication that uses eye tracking, that uses residual puff and blow function for people. And that's been tested in hundreds of thousands of patients. So why is it that you're seeing finely articulated robotic arms and not control of existing assistive technologies where rehab facilities and physicians that do this with patients every day could actually use these devices. Well, it's easy enough to get back-end control of these, right? Some of these companies will release their APIs. You know, for folks who want to think about what the barriers are to neurotechnologies, I think this is a reasonable paper. There's going to be QR codes on a number of these, but you actually need to consider what does it mean to get adopted? What would it mean for brain-computer interfaces to get adopted? How easy does it have to be for neurosurgeons to put in, but more importantly for neurologists to take care of them over the long term, right? There has been, I think, a really good example of a practical scalable device from Nick Ramsey's group in Utrecht. This was a device that used the Medtronic PC+S device, gave people 1 to 2 dimensions of control, and it actually helped patients with ALS. They gave them open use from the start so that the patients could use it at home. And as the patients became more locked in, and there's been several now, the use of the device goes up. And they've reported how those signals change over time. So there's principles that they're learning that have to do with very basic signals that they've been publishing. I think this is a beautiful thing, and I think these should be the things that are getting the attention. I'm going to try to tell you a little bit about the efforts that I've been lucky to be a part of over the course of my career that maybe have some general principles about what these are. So not ethical principles, but general principles for what kinds of things we need to create brain-computer interfaces. Okay, so I think a lot of the studies, my point is, are doing this kind of thing where you have scattered observations that are all flashy independently. Maybe these should have been gold-plated Legos. But what we need instead is this, where we develop principles and we try to stack the principles that we have in ways that are intuitive. So each study, each investment that the public makes with their tax dollars and grant dollars becomes something that helps with future technologies. So a question we could ask is, well, what are the electrophysiological source signals that we can leverage? Okay, so right now, the types of electrodes that we put in and the types of signals that we measure really boil down to a few basic things, and we should think about for the applications that we're interested in what these are, right? So we have penetrating electrodes that are macro scale. DBS electrodes are an example of this. This is a stereo EEG electrode. And we measure voltage, and that voltage will average over some amount of a population of neurons. We may have microelectrodes that we put in. I do this a lot with my deep brain stimulation surgeries. Of course, I'm lucky enough to have followed one of the pioneers in understanding single unit microelectrophysiology and its use in DBS surgery. Dr. Vitek really is the pioneer for this. And that captures very high fidelity activity in the thalamus or in the basal ganglia that we use to help guide our larger electrodes because of the specificity that those have. And so from this voltage, we can do things like we can extract rhythms. So these are oscillations that are there. We can see usually decreases in the amplitude of these rhythms when that brain area is engaged for a variety of reasons. We can have event-locked raw voltage deflections. So these can come from visual stimuli, auditory stimuli. They can come from surprise. And they can also come from electrical stimulation. And so here in the front row, we have a large number of people from Dr. Hermes' lab who will be joining us later. And that's really what I'm proud of, what we're pioneering right now at Mayo. There are also what are called broadband spectral changes, and I'll show you a little bit more about that later. But these are measured sort of at the macro scale. And then we have action potentials. And action potentials are really where the fanciest stuff is shown. But we need to understand that I think for a toolkit of electrophysiology, these are the most robust phenomena that we can find. Each signal feature reflects a different aspect of cortical computation. Each is best captured at a different scale. And individual brain-computer interface constructs may benefit from a synthesis of these. So it's not that one's the best. It's that you may want to, let's say you're using action potentials where the baseline firing rates of neurons, the baseline action potential rates, scale as brain state changes, as you're more or less drowsy. And you're not going to capture more or less drowsy very easily with action potentials, but you are going to capture it with oscillations in the brain, okay? And so we can modulate how we decode one feature from the measurement we make from a different feature. I'm going to be talking a lot about electrocorticography. That's what I had a lot of access to as a graduate student. And it's what I think a lot of brain-computer interfaces will have moving forward. And a lot of the principles that we see for electrocorticography generalize to depth electrodes, measured from DBS or stereo EEG. And here, what we think about is an electrode on the surface of the brain. We measure some physiology due primarily, probably, to pyramidal neurons and cortex generating differences in charge between superficial and deep cortical layers. And we found that these generate stochastic, or what look like stochastic signals, or random walks in the time series of voltage that we measure, and these obey something called the power law. We also, as I showed you before, have evoked potentials, and we have oscillations that we can measure at the scale of these electrocorticography electrodes. And so as we think about these source signals, we want to ask ourselves, well, what are the features of each and what are the limitations of each? And I'm just showing that one feature and one limitation of each, but obviously there's a whole set for each of them. So for oscillations, they're widely present spatially, okay? They're now usable as a biomarker with closed-loop devices from Medtronic and from NeuroPace. But they tend to be functionally non-specific, okay? So I'm going to show you some examples of how well they resolve somatotopy. It's not great, but they're spatially very widely present. We can capture them, and we can measure basic behavioral changes. These event-locked voltage deflections that we get are temporally very precise, but they require a supervised paradigm. You need to know when somebody saw something new to know if the thing that they saw that was new registered a unique event inside their brain. So the way you approach it has to be supervised. These broadband spectral changes are very functionally precise, but you are capturing things that have relatively low signal to noise. So if you're trying to design a closed-loop brain-computer interface device, you have to have pretty good hardware. Your signal to noise has to be pretty good compared to what you would need to capture oscillations or evoke potentials. And then there's action potentials. So an example feature of these are that's the finest resolution that we know how to get of the brain's computation. We're not usually looking at least in vivo, not looking at channels or anything like that. And by channel, ion channels, that kind of thing. But people often tout the fact that this might have high temporal fidelity, but because these are intrinsically stochastic, right? They come at least to first order, they're Poisson distributed. You have to integrate over time to get any kind of temporal information from them. So they're not temporally precise, and you can do well maybe by averaging over hundreds of them, but if you have hundreds of channels that you can measure from, I would propose that maybe some of the larger scale electrodes with these larger features would be more relevant. So this is an electrocorticography array. The way we get to do this is we're working with epilepsy patients, and I'm fortunate enough to be able to work with a lot of epilepsy patients. We implant these in order to localize where patient seizures start from. But we've actually mostly transitioned away from electrocorticography towards something called stereo EEG, where I make little pinholes in the skull, put bolts in, and then advance wires through them with about 1 to 2 millimeters of precision, maybe 3 millimeters of precision in some case. So a little worse than the precision that we get with DBS, but actually quite high fidelity in order to, let's say, understand the physiology and relevance of specific thalamic nuclei for a patient's seizure network. And every time we think about a scale we're measuring from, we want to sort of think about, well, what does this reflect? So each one of these electrocorticography electrodes is about 500,000 neurons. We're able to work with patients who are implanted otherwise, so we're not introducing any extra burden, but of course we only get the electrodes where they're needed clinically, not where we would like to measure from. This is also true of stereo EEG. Some basic phenomena that we see with these are if you measure from functional, like from primary motor cortex, precentral gyrus, you have patients stare at a blank screen. You can measure power as a function of frequency and the voltage that you measure, the power spectral density, and you can give them some cue to engage in some behavior, and if you're overhand area of the brain and give them a cue to do that, you actually see well-described and well-characterized phenomena. The first is that you have spatially focal changes at high frequency that reflect increases in power. Then you have spatially very broad decreases in power in these low-frequency oscillations. And this is now from 2007, so 18 years ago when we did this, so it's belying my age, and I was already almost 30 when we did this, but we thought that actually, what we were seeing at higher frequencies wasn't high-frequency oscillations at all. What we were seeing were what are called broadband changes or power law changes in the power spectrum. And that's important because we can then try to understand something about the underlying statistics of these. When we saw that you had high spatial fidelity with the captured at the high frequencies, we said, well, maybe we can map. This was also 18 years ago. You'll notice the lack of spots on my hands in a second here. But we could do real-time brain mapping with this for functional mapping. And companies like GTech have made brain mapping devices that you can use during awake craniotomies in real time for this. And so we can rapidly map out function on the brain surface using these high-frequency changes. And so in order to ask whether this hypothesis is correct, we use this little trick where we look at different power ranges in the power spectral density. And I had patients move their thumb and index finger separately. Their thumb and each of their fingers separately. What I didn't know at the time was that most of the primate physiologists would say if you measure from any area within the hand area of the brain, you're going to get representation for all of the different fingers of the brain. I only knew the classic Penfield story, which is that individual fingers are in different spots of the brain. So I thought, well, if you have oscillations that are widespread and you have these high-frequency changes that are spatially focal, if we can generate different variants in those things spatially, maybe we can rip apart the power spectral density using a decomposition. And it turned out that actually you can do that. So it seems that for averaging, there really is somatotopy and I'll show you some examples of this in the human motor cortex. But what we could do is if you imagine just taking power at a high and low frequency, you can plot them on the X and Y axis, and you see basically with going from rest, which is the black dots here, to movement of paired finger, you have decrease in low frequency power, but increase in high frequency power. And if you have other fingers that are not the paired fingers, this was, I think, a thumb-specific site, you just have mostly a decrease in low frequency power without a corresponding increase in high frequency power. And if you think that there are multiple phenomena overlapping in the power spectrum, I show this for 2 power bands, you can rotate your axes here. And what that amounts to is a principal component decomposition. You use the variance in the 2 signals to rip them apart. And this becomes relevant because if you do this instead of at 2 power bands, you can do this at single individual frequency bins. You now see that the most significant change is actually pretty even across all frequencies. And when you plot that back into your power spectral density, that corresponds to an increase in all frequencies separate from oscillations. So this really is this broadband spectral change that you have here, meaning it's changing across all frequencies. You don't see it at low frequencies because you have oscillations there, but you can use tricks to rip it apart and away from it. And I'll get to why that's important in a moment. And so if we measure our electrocorticography signal while we have patients move their fingers with the data glove, we get basically decreases in low frequency power here in the classic motor oscillations between about 12 and 20 hertz, measured from the precentral gyrus. They're spatially very broad, okay? So blue here is a decrease in power, these low frequency oscillations. But if we use some signal processing tricks, we can extract that broadband, and you see that here in pink, and that's very highly specific, not just for sort of the times that the thumb moved over an average time, but actually individual movements of the thumb are captured just from this single electrode that we've looked at here and not the others. So this tells you that you have a way of capturing relatively high fidelity temporal information that is spatially very focal. And the reason why we get that is that compared to, let's say, single units where you have to average over many, many units in order to get this kind of temporal precision, is that we are averaging over many, many units. We have about 500,000 units underneath the electrodes of this scale. And so what we can see is that, again, this is just a different patient. This red dot means it's very significant increase in broadband spectral change. So that's including both low and high frequencies, but you've stripped away the variance of the low frequency oscillations in the thumb area. But then for these beta oscillations, you have widespread changes across a much broader portion of the area in front of the in front of the central sulcus that is pre and primary motor areas, okay? And if we look at different fingers, we now have representation at a single site for the index and little finger that are 1 centimeter away from where the thumb was. And we can quantify the overlap between them. We see that these beta rhythms or oscillations between 12 and 20 hertz are overlapping for all the fingers, and that may explain why you see some units, regardless of where you look in hand area, that are tuned to specific fingers. But when you look at this broadband spectral change, they're not overlapping. And if you look at time-to-resolve versions of this, you have electrodes that capture the fine temporal dynamics of movement of the thumb, index finger, or little finger. This is at the scale of about 5 square millimeters underneath each one of these electrodes. And we're putting them for clinically spaced at about a centimeter from one another, so it's about 5% of the cortex that we're sampling from, and within that 5%, you see these domains. So this principle that we've learned is we can leverage these different signals as different phenomena that we might use to control brain-computer interfaces. So if we use this for feedback, what happens? Well, it turns out that if you have somebody move their hand, as I showed you before, you get characteristic changes. But if you have them imagine doing the same thing, you get just a weaker version of those same changes, okay? And we can capture low and high frequencies. Here, these high frequencies, it's not that that's a special frequency range. It's just that that's where we don't have the oscillations, and we're above where our noise floor of our amplifiers kick in, okay? So we're in a happy range to capture this phenomena that's really spread across the whole frequency domain. And we see that imagery very highly correlates with movement for paired modalities, and that's what you're seeing here. So hand imagery looks like hand movement, tongue imagery looks like tongue movement, and they're not overlapping with one another. We can then just pair this simply to the movement of a cursor on a screen, so we capture in individual electrodes, high frequencies. These are not sophisticated algorithms, but it does let us look at the simple properties of the populations of neurons that we're looking at, so we can do two-dimensional cursor control. This is 15 years old. It wasn't necessarily even that sophisticated for 15 years ago. It's certainly not sophisticated now. But you can, within a few minutes, have somebody controlling a cursor in 2 dimensions, just using 2 electrodes, one from the hand, one from the tongue area, and after about only 10 minutes of training with simple linear mapping between the velocity of the cursor and the power that you measure at these sites in that high-frequency range that captures broadband spectral change. And so we can look at learning in this, and if we compare the power in this higher frequency capturing of broadband in upper and lower targets in a one-dimensional brain-computer interface, we see that over the course of several experimental runs, so this is 2 minutes, 4 minutes, this is how much time they're actually participating, they can control the cursor with high signal fidelity, getting 94% accuracy after, and that's pretty characteristic also of what we get with stereo EEG that I'll show you, after just a few minutes of training. And what we find is that after feedback, actually, if we look at the amount of activation or the amount of power change, feedback in the site of feedback, the amount of power that the brain generates in the signal there is larger than what you have during both overt and imagined movement, okay? And this was for one patient, but these are for several other patients. And so as a principle, that tells us that, well, when you give feedback to a brain area, and for single units, a guy named Eb Fetz, who's sort of the father of brain-computer interfacing, showed that you could do this in 1969 in a beautiful science paper with operantly conditioned single neurons. But we can do this just over a few minutes of feedback. We can generate activity that's more. Well, I think that's useful to know. And that's useful for why macro electrodes here might be important. And if you wanted to have somebody who learns what their own mapping should be in the morning before controlling a closed-loop device, you can do it with these kinds of electrodes without having a whole team of people in that person's house decoding, from 100 different electrodes. It's just from a single one. Okay? So we could say, well, is scale important here? Is the scale of the electrodes important? Well, I think that's something that we should be asking ourselves. Not what's the best scale, but how does the signals that we measure, how do they vary as a function of electrode sizes? Well, actually, I'll be showing some recent stuff, and this is a question that scientist Rosanna Esteler and I have been working on together, as well as the other individuals in my lab. But if we think about how these broadband spectral changes might emerge, we can come up with a toy model, right? All models are wrong, but some are useful. Well, this is a toy model that generates broadband spectral change. If you consider one pyramidal neuron in a population of neurons, and it gets stochastic inputs of action potentials, and each of these gets filtered by the shape of the postsynaptic current, and you have current that flows from superficial cortical layers to deeper cortical layers with charge flowing out as it goes, you can generate synthetic time series, and you see the same types of broadband spectral change in synthetic time series that we see with our actual data. And so the reason why this is important is as you're averaging over a larger number of neurons, the spatial scale, if this is true, of population size that you're measuring in the electrocorticography signal should scale with electrode size. And this is not true for oscillations, and I'll leave that to questions that you might have after in the interest of time, but it is true with with these electrodes that we measure from. So this is just showing you the specificity that we had from from a thumb and an index finger specific electrocorticography electrode at the standard scale of 5 square millimeters. But if we do the same with high density electrode arrays, and this is work that I did, but still a publication for from 6 years ago when I was a postdoc with Nick Ramsey. Everyone's bad, so although Michael, you got to finish your PhD. What we see is that the ability to resolve somatotopy is actually worse with smaller electrodes. So we gain something by intrinsic averaging. So having larger electrodes, as long as they sit within that somatotopic specificity, actually gives us computational power. I think that's useful to know. And I don't have that all worked out, but it's a problem that I think is useful for us to work on. And we can start to generate what we think are candidate scales using some proxies. One nice proxy is fMRI, although in a sense it's a more blurred out and sloppy version of what we would want to get electrophysiologically because it's a metabolic reflection. But it is a useful signal, and we do find that if we look at bold signal change and we compare that to these higher frequencies, you see that there is a tight correlation in a primary area of the brain, at least during movement, between the bold signal and changes in the power spectrum. And so what we can do with that is we can try to model optimal grid configurations with 7T fMRI. So this is actually also from Nick Ramsey's lab and Dora Hermes' lab, but the student at the time who did it was Max van de Boom, who's a Dutch guy. And what he did is he took these 7T maps, and I also realized talking to you guys about 7T is not doing you the justice you deserve, because University of Minnesota, is the CMRR here is probably the best imaging center in the world. But anyway, this could be something to try, let's say in collaboration with another grant we could do together. But we can model different grid sizes and try to come up with optimal grid configurations that we might have for a particular patient before we would, let's say, implant them for a brain-computer interface. And what we can also do is we can look at different electrode scales during DBS surgery. If you guys have if anyone has any interest in doing this, this is a custom array here that we built with Cortec that we paid them to make for us, but it can just be ordered. Getting around this whole idea of exclusivity. Look at 10 minutes. Good. This is not exclusive. So if you guys want to do this and you want to measure from different scales of electrodes, we paid for them to make this. You can just order it. I think this is really nice. We can slide strips of electrodes back, let's say, in my case, over primary motor cortex, and you pull this strip back and repeat tasks to look at different scales of electrodes during DBS surgery. We're not implanting grids that often anymore, but doing this during DBS is validated by Phil Starr in San Francisco with 300 patients showing that it's safe. So we had 300 patients worth of data showing that you could take at least clinical strips, and this has the same footprint as that, and measure from them. So we can actually do this with our DBS patients, and we're starting to see some interesting results. This is what I'm working with Dr. Esteler on. So basically, different macroscale electrode sizes differentially capture local representation. So there's no one best electrode scale. And actually, if you look at oscillations, larger electrodes average over even more cortex. And there may be benefits actually sometimes in capturing oscillations for being outside of the head altogether and getting that spatial averaging that happens as you pull away from cortex using, let's say, subgaleal electrodes combined with DBS. Okay, another principle we might want to ask is we have these different features. Do they carry different types of decodable information? Are oscillations different from, I've shown you oscillations are different from broadband, but are evoked potentials different from broadband changes? We have fMRI bold signals, and like I showed you before, we can place electrodes based on that, and there's a well-known phenomena of different categories being represented in different brain areas in the inferotemporal cortex. I think this is really well established by Nancy Kanwisher in the 90s. But we did this simple visual experiment a couple decades ago where we just show people basic pictures of faces and houses in order to get them to attend. We said, tell me when you see the upside-down house. I'm sure you guys could see it. That's the experiment we had them do. And we can then measure the voltage during that, and we can also extract these broadband changes like I showed you. We can look at the changes that we see here in the raw voltage when we see pictures of faces. This is Dr. Hermes here, and then we also have pictures of houses here, and we can see the times when we delivered that. And we can look at the raw voltage deflections that we get there. And then we can also capture these broadband spectral changes. This is a measure of frequency and time, just showing that after the stimulus, we have a site here that's specific for faces. And we can do eyeball classification. So if we do broadband extraction here, you can see the times this spikes up are basically only at the times when faces were shown. And we can ask ourselves, is there different information here? So I can say, well, if I just look at the time, knowing ahead of time when images were shown, ERBB here is just the broadband that we get, the broadband time course that we get in response to showing these images. This is our classification accuracy of faces versus houses. And what we see is that basically broadband seems to do better in most of these are different patients along the bottom here. And this is our percent correct. You know, with the broadband, we're above 90% in almost all of them. But actually, when we put them together, it increases our accuracy. There seems to be something complementary about just if we predefine what's happening, about our ability to decode here, okay? But we can also look at this in real-time decoding, and we can, you know, say, well, when the patient sees this image, we can measure the voltages, extract broadband, and get the raw voltage deflections, and then we can project that into a feature space here. And I'm going to play some sounds. I don't know if the sounds are working. But we can, in this feature space, we can then apply machine learning algorithms. I actually think linear, which is just a simple Fisher LDA in real time. Linear algorithms seem to work the easiest with these types of electrophysiologic data. But by putting these in a feature space together, we can then try to decode and ask how our decoding changes as we remove one feature or another, or we look at them together and ask whether this information complements each other or not. Okay, so here we're decoding instantaneous brain state. And there's beautiful data sets that we've been able to generate at Mayo with collaborators here with Kendrick Kay, who's developed a really nice natural scenes data set that he's looking at with fMRI here at University of Minnesota. We're doing something similar, not just with faces and houses, but with natural scenes here at, down at Mayo. And Zishan, one of our graduate students, has a nice publication that should be going up on archive soon, if it hasn't already, about these data. But what we find is actually that if we look at this spontaneous data stream, we can compare accuracy and capturing it spontaneously. And then we can look at our error in terms of false predictions that are incorrect, and also our error in time. And what we see here in red is that if you combine the broadband spectral changes and the evoked potentials together, you have fewer false predictions of having seen something, you have higher accuracy, and you have better temporal fidelity. So that's a general principle that I almost got a paper rejected by having the title be, at this point I was thinking, well, the most descriptive titles you could have would be most likely to get discovered when people do internet searches. But this was the basic phenomena that we measured. Basically, you have complementary information in these two types of signals. And so it's not that one's better than the other, or you should use one or the other in your algorithms. It's you should capture these independently and synthesize them together, okay? I'll be good in about 5 minutes. Is that okay? So we've made a transition to stereo EEG clinically. This has some advantages and drawbacks compared with electrocorticography. I like being able to work with my patients. They don't have any extra burden. But actually, it may be that the way the brain reacts in terms of encapsulation may be better with stereo EEG, and the stability of signals may be better with depth electrodes. So I don't need stereo EEG. I guess in this case I would need something more like DBS electrodes than it is with surface electrodes. But basically, we now can't do very well at capturing functional domains with this, and even in the future we won't be able to as well, presumably because of the way we anchor these. I mean, you could have large penetrating macro electrodes and a bunch of them together, but we may start to see deficits in patients because of the trauma we induce by doing that. But what we can do is we can capture deep structures in the thalamus, we can capture deep structures in the basal ganglia, and we can capture multiple points in hierarchy, right? So we can not just get primary motor cortex, but we can get supplementary motor area, we can get thalamic motor areas in tandem with the cortical surface. And this may be the way we should be thinking about generating new features for brain-computer interfaces. And so if we do the same types of basic experiments I showed you earlier for electrocorticography with stereo EEG, we see the same broadband spectral changes, we see the same decreases in oscillations, and we can start to ask some basic questions. So do our ideas that we had about the motor homunculus extend down into the depths of the central sulcus? This would be important for, let's say, brain-computer interface features. And what we found was something really cool. That is, first of all, with these white arrows here, these are axial cross-sections of change that we get in movement compared with rest. Basically, you do see hand, foot, and tongue areas in the expected places. In addition to that, deep in the depths of the central sulcus, on the precentral gyrus, so classic Brodmann area 4 that we always thought should be somatotopically organized, at least at this scale, you have areas that are, in fact, somatotopically nonspecific. They have shared representation. And this wasn't known. So it's kind of nice. And so deep in the depths of the central sulcus, even though you have foot, hand, and tongue areas, you also have these intermixed areas that you'll see pop up here in red that have some kind of motor association role. Okay? And so that turns out to be a useful principle. As we start probing the depths of the central sulcus, probing the depths of the brain in a variety of different places, we may discover different elements of the motor hierarchy that we can leverage for brain-computer interfacing. I'm going to skip past that. And then the last thing that I think is important is if we run brain-computer interfaces with stereo EEG in the same fashion that I showed you before, we're actually able to do the same types of experiments that I showed you that were relatively rudimentary 15 years ago. Well, they're even more rudimentary now, right, with people decoding speech and doing other things. But maybe we can discover some useful principles from this, okay? And we now have about 25 patients where we've had them do these one-dimensional brain-computer interfaces. And the purpose here isn't to demonstrate, I mean, this is as simple of a task as you can get. You can actually do this with EEG. But the purpose here isn't to say, hey, this is what a brain-computer interface is going to look like. Hey, Alec. The purpose here is to ask, well, what happens to this network of brain areas as we give feedback to a primary motor site? Okay? This is where my resident wrote my initials. If any of you go into surgery, make sure you see the patient preoperatively yourself. You can put your initials over their initials. I try to do that every case. My surgical protege and grad student, Dr. Karazutis, knows this well. Basically, when we do motor imagery, just like I showed you before, we can drive brain-computer interfacing off of that, and we can learn over the same time scales, over 10 minutes or so, to control one-dimensional brain-computer interfaces. We've done this now with, this was about a year ago, so I think we have more patients now. It's in the mid-20s. But we are able to control brain-computer interfaces robustly from early motor areas with implanted depth electrodes here. Now, that's not what's interesting. What's interesting is that when we do that and we compare movement to imagery, well, we might expect it when you're not performing actual movements, you would see engagement of premotor areas preferentially over primary motor areas. We do see that. But we also see areas, such as the ones you see down here in the vicinity of parietal reach region, which are differentially and preferentially engaged during feedback and not engaged during reinforcement of primary motor cortex. So that tells you that feedback uniquely engages brain regions outside of the site being reinforced, okay? So that's kind of a neat principle we can learn from basic brain-computer interfaces, okay? And our goal here is to try to develop principles, not that make it so we're going to be the place that solves brain-computer interfacing and get what Beneby, which I believe Beneby deserves, which is a Nobel Prize. That's not the kind of thing we're doing. We're doing the kind of thing that we think should help other labs so that they can develop more sophisticated devices with high numbers of channels, and we're doing that in the context of just working with patients that we already have. An interesting question we want to look at in the future, so the sound won't work here, but basically we can couple this one-dimensional control to this is a classic game called Galaga, which I played on a machine when I was younger, and most people in here probably don't know what this is, but this is what video games used to look like. And one thing that's important is what happens as we enrich the feedback. Are signals stable under that situation? So these are the types of questions that we think are important for a future principal to ask. Okay, so do I have 2 or 3 minutes to rant? 2? 1, okay. Okay, so we need end effectors, and end effectors means devices that the patients actually interact with, that are patient-centered and leverage existing assistive technologies. That's something we want to do, okay? I think that's what most people should be doing. They should be saying what's actually needed by these patients and use things that have been used by hundreds of thousands of patients who have residual eye function residual motor function. It may not be the best idea to have these high-density arrays anyway. If you want somebody to know that they've seen a face in a visual prosthetic and that it's a particular part of their visual field, trying to reconstruct all of vision in V1 doesn't make a lot of sense based on what we know about how the brain is. You should be stimulating face area. You can do that with a single electrode at the same time as you generate a phosphine, coincidentally, in early primary motor cortex, at the same time as you generate this perception of motion in MT. That's 3 electrodes. Now, of course, you need a wider set of electrodes than that, but you're generating what should be a stable percept, leveraging the distributed architecture in the brain. You're not generating a complete reconstruction of a face in primary motor cortex, which people are still trying to do, but we already know empirically doesn't work because people have been implanted with this. Okay? This is what it looks like when I take paddles out. So even if you have surface electrodes, you need to know that when you take these paddles out, this is after just 4 years. This is a patient who was actually implanted here at University of Minnesota. Initially had a great response, but over time had less and less utility out of it. And we were able to, after discussions with our neurosurgical colleagues here, able to go in. But in order to remove this device, I had to do a large craniotomy, right? So this gets slid in through a burr hole. If you slide these strips in through a burr hole, you're not taking them back out through a burr hole later. Furthermore, the signals that you measure now are going through this amount of gliosis, so your electrode sizes actually should still give you a robust signal after a couple millimeters of gliosis if you have electrodes sitting on the surface. If you have the Utah arrays, that several centimeters pulls you out. You now, whatever signal fidelity you had in the beginning is gone in a couple years. Okay, and the last thing is that I thought I should end on something really high tech. So this is a drawing here, okay? So your control signals vary as a function of shift in state in the brain. As you get more drowsy, your ability to resolve your control signals is going to change over time. I'm lucky enough to be funded by, okay, so I royally messed up. I also should have the Minnesota partnership grant here that I had with Dr. Johnson. I'm sorry. Funding is important, and we're lucky to be funded by the state of Minnesota. I'm very grateful for that, and I'm also very grateful to be able to collaborate with the University of Minnesota. I'm part of a large team. These are the folks on the team that worked on the brain-computer interface projects that we talked about, but there's a lot of other great work, especially by people in this room who drove up from Rochester, so I'm not neglecting that. It just wasn't the BCI stuff. Thank you, guys. Appreciate it.
Thank you, Dr. Miller. Maybe we could have time for one quick question before we move on to session 1.
Can Dr. Vitek ask a question? If he has a question. If I'm going to have one person.
It was prophetic, I guess. Have you followed the work by Nico Dosenbach out of WashU with these inter-effector regions mixed in with the effector regions?
Yeah, sure.
And
hey, and it's my first keynote, so I'm going to talk to you about that.
Okay, and how do you kind of pull that into the work you're doing?
So there's co-authors common to both papers. We put our stuff up at the same time as them. We thought there was overlap. We said, hey, we should talk. So scientific collaboration with the University of Minnesota has been how it's been. We said, it's been how it should be. We actually talked to them. We said, hey, we think there may be some similarity between this. Why don't we put them out as paired papers? And they said, sure, that would be great. We had a meeting with them online. We showed them where we're at. Michael actually gave a talk at WashU, and then they went to their editor and got their paper out a month before ours, when we both submitted journals at the same time and let our editors know. So first of all, I think they're bad citizens scientifically. And so I have a keynote. I'd love to be able to rant about that. So I'm sorry for ranting, but hey, that's what you get that for. The second thing is that I do think that these areas integrate information over a wide range of somatotopic specificities, and something that I think was really good about the work that they did that seems to be something we should explore more is this idea that it may be relays between thalamic nuclei that we've classically thought of as having output to basal ganglia, like the centromedian nucleus, coordinating activity in cortex. And so that's something that I think needs to be explored electrophysiologically and that we're doing. So I do think that there are elements of that were great. I'm bitter, but you still have to respect the science because I think it was good science.
That's okay. Thanks for the talk.
Thank you. Yeah.
Great. Thank you, Dr. Miller.